Welcome back, my dear students. Today we will briefly cover the origin and classification of metazoa, what metazoa are, the key classification criteria, that is levels of organization, germ layers, coelom, symmetry, segmentation, protostome versus deuterostome, etc. Major origin theories, that is Syncytial theory, Colonial theory, polyphyletic theory, and modern quanoflagellate or monophyletic evidence, and types and evolution of symmetry from asymmetry to bilateral, including secondary radiality in echinoderms, with quick examples from Porifera to Chordata and a few bite-sized exam mnemonics. So let's go step by step and make this topic simple and memorable for your exam purpose. Introduction to Metazoa. Metazoans are basically include all multicellular animals excluding the unicellular protozoans. They are multicellular, we know. They are eukaryotic, they heterotrophic organisms. All of them develop from a blastula stage during embryonic development. Exception Porifera lacks true tissues, but they are included in Metazoa. Key features of the Metazoa are cellular differentiation and division of liver, embryonic development, cleavage, blastula, gastrulation, huge morphological diversity ranging from sponges to human. Classification of Metazoa phyla based on morphology and development. Levels of organization, cellular level, loose cell aggregates, example Porifera, tissue level, true tissues like Nidiria, Ctenophora, organ level, organs are formed, for example Platyhelminthes, organ system level, that is complex systems found in arthropoda and chordata. Body symmetry, asymmetry, there is no definite symmetry which is found in case of poriferan animals. Radial symmetry, body can be divisible along in many planes which is found in Nigeria, Ctenophora, adult Echinodermata, etc. Bilateral symmetry, only one plane divides into two equal halves, most phyla from Platyhelminthes to Chordata. Germ layers, it can be diploblastic or triploblastic. It has two layers, that is ectoderm and endoderm, which are found in Nigeria and Ctenophora. Triploblastic, there are three layers, ectoderm, mesoderm and endoderm, which is found among the higher metazoans. Body cavity or coelom. Acylomate, there is no body cavity, just like Platyhelminthes. Pseudocoelomate, there is body cavity but not fully lined by mesoderm. It is found in Nematoda. Coelomate, there is true coelom which is fully lined by mesoderm, found in Annelida, Mollusca, Arthropoda, Echinodermata and Chordata. Now you should study the developmental patterns of the metazoans. Protostomia, where blastopore forms mouth, spiral and determinate cleavage is found. When there is zygosily, they are called as protostome animals. Examples include Annelida, Mollusca, Arthropoda. And deuterostomes are where the blastopore forms anus and radial and intermediate cleavage is found. The enterocily, and then their examples include Echinodermata, Hemichordata, and Chordata. Segmentation or metamerism, that is absent in mollusca or in nematoda, but they are present in annelida, arthropoda and chordata. Skeleton, hydrostatic skeleton is found in Nigeria and annelida. Exoskeleton is found in arthropoda and mollusca. Endoskeleton is found in echinodermata, chordata. Circulatory system, it is absent in porifera or Nigeria, but open circulatory system is found in arthropoda, most mollusca, but closed circulatory system is found in other animals like Annelida, Chordata, Echinodermata, etc. Hypothesis of metazoan origin. Syncytial theory, which was proposed by Haeckel, Haji and Hansen in 1958. It suggests that metazoans evolved from a multinucleated ciliate-like syncytia. Their cellularization led to primitive acyl flatworms like Turbellaria. Their internal bilateral gave rise to radial cnidarians, that is their secondary radial support. The similarities between ciliates and flatworms support this syncytial theory. Criticism: Acilla are fully cellular, ciliate macronuclei absent in metazoans, and their embryonic cleavage differs. Colonial flagellate theory or gastria theory: Haeckel, Butstrelli, Langster, Metchnikoff, Hyman, etc. They were proposed this theory. Metazoans, according to this theory, metazoans originated from colonial flagellates like guanoflagellates. Their colony form the hollow sphere or blastia. After invagination, it will form a gastria, that is prototype metazoan. Support: blastula and gastrula stages of embryos, coanocyte resemblance, flagellated sperm. Criticism: ancestral volvocyte flagellates were plant-like, and some differences in development. Status: most widely accepted theory. They are also supported by modern molecular evidence. Polyphyletic theory, that is the suggest different polyphyletic theory suggests different animal groups evolved independently from different protozoans. Examples include sponges from flagellates, flatworms from ciliates, etc. Criticism: this theory fails to explain common embryological patterns of all metazoans. Other theories are plasmodial theory, which was originated from multinucleated amoeboid ancestors, and coanoflagellate lineage hypothesis, or modern molecular theory, which supports the monophyletic origin of coanoflagellates, reinforcing the colonial flagellate ideal. Conclusion: the colonial flagellate theory or gastria theory is still the most accepted explanation, now strongly supported by molecular data showing that coanoflagellates are the closest relatives of the metazoans. Symmetry in metazoa. Types of symmetry. Asymmetry, that is, there is no definite symmetry which is seen in sponges. Spherical symmetry, they are rare. Any plane divide body equally, that is seen in some protozoans like volvox. Radial symmetry, their body can be divided into equal halves along oral aboral axis, that is seen in Nigeria, Ctenophora, adult echinodermata, and they are adaptive for sessile or floating life. Bilateral symmetry, that is mixture of radial and bilateral, that is seen in Ctenophora. Bilateral symmetry includes body can be divisible into right and left halves along sagittal plane, that is seen in most higher metazoans. They are adaptive for cephalization, directed movement, and complex nervous system. Evolution of symmetry: asymmetry, that is found in sponges, then radial symmetry found in sessile diploblast, after that bilateral symmetry, which is found in active triploblast. So the bilateral symmetry enabled cephalization and efficient locomotion. Secondary radial symmetry evolved in adult echinoderms from bilateral larval forms. This image or the chart shows a flowchart of the classification of kingdom Animalia beginning with Protozoa and Metazoa. It highlights major groups such as Porifera, Nigeria, Ctenophora, Platyhelminthes, Nematohelminthes, Annelida, Arthropoda, Mollusca, Echinodermata, Hemichordata, and Chordata, further dividing chordata into cephalochordata, urochordata, and vertebrata. The vertebrata are then classified into cyclostomata, agnatha, and gnathostomata, which lead to fishes, amphibians, reptiles, birds, and mammals. So, in today's lecture, we explored the origin and different classification aspects of metazoa, looked into the major theories of metazoan evolution, and understood the different types of symmetry in animals along with their evolutionary and adaptive significance. Remember, this concept form the foundation of animal diversity, linking simple organisms like sponges to complex vertebrates. If this video helped you, don't forget to like, share, and subscribe to stay updated with more easy and exam oriented biology lessons. Thank you.